Chromosomes are located inside the nucleus of animal and plant cells. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. Passed from parents to offspring, DNA contains the specific information that makes each type of living creature unique. Chromosomes are not visible in the cell's nucleus, not even under a microscope when the cell is not dividing. However, the DNA that makes up chromosomes becomes more tightly packed during cell division and is then visible under a microscope. Most of what researchers know about chromosomes was learned by observing chromosomes during cell division. In this session, we will focus on special type of chromosome like Polytene chromosome and Lampbrush chromosome. Polytene chromosomes were first observed by E.G. Balbiani in 1881 in the larval salivary gland of Chironomus midges and were studied by Korschelt and Cornet in 1884. The hereditary nature of these structures was not confirmed until they were studied in Drosophila melanogaster in the early 1930s by Emil Hertzsmil and Hans Bohr. The name Polytene bicolor was assigned to these chromosomes due to their large size showing numerous strands. Polytene chromosomes are giant chromosomes common to many dipteran flies. As the name suggests, poly means many and teen stands for strands. They begin as normal chromosomes, but through repeated rounds of DNA replication without any cell division called endoreplication, they become large banded chromosomes. For unknown reason, the centromeric regions of the chromosomes do not endoreplicate very well. As a result, the centromeres of all the chromosomes bundle together in a mass called the chromocenter. Polytene chromosomes are specific interphase chromosomes consisting of thousands of deoxyribonucleic acid strands. For this reason, they are very large and display characteristic band-interband morphology. Organs containing cells with polytene chromosomes are, as a rule, involved in intense secretory functions accomplished during short time against a background of rapid growth. Polytene chromosomes allow a specific narrow region to be dissected out with a micromanipulator, and a library of DNA clones can be derived from this region. The polytene chromosomes are present in the salivary glands of the fruit fly Drosophila melanogaster. Formation of these giant chromosomes involve specialized cells which undergo repeated rounds of DNA replication without cell division to increase cell volume, and due to this multiple round of replication, many sister chromatids produced remain fused together and form polytene. Besides salivary glands, the polytene chromosomes also occur in rectal epithelium and Malpighian tubules. The size of the nuclei in the salivary gland cells are about 25 micrometer in diameter, which is much larger than the normal ones and consist of giant chromosomes which are bigger by 50 to 200 times as compared to the normal chromosomes. According to an estimate, the polytene chromosomes have 1000 times more DNA than the normal somatic chromosome. Polytene chromosomes develop from the chromosomes of diploid nuclei by successive duplication of each chromosome element. The characteristics light and dark banding patterns of the polytene chromosomes of Drosophila melanogaster were sketched in 1935 by Calvin B. Bridges that can be used to identify chromosomal rearrangements and deletions. Dark banding frequently corresponds to inactive chromatin, whereas light banding is usually found at areas with higher transcriptional activity. These banding patterns provide an excellent visualization of transcriptionally active chromatin and general chromatin structure. For example, the Polytene chromosomes in Drosophila have been used to support the theory of genomic equivalence, which states that all of the cells in the body maintain the same genome. Polytene chromosomes consist sites for RNA transcription in the form of diffused uncoiled regions known as chromosome puffs. Balbiani ring is the largest chromosome puff. The length of Polytene chromosomes is about 200 micrometer, and this enlargement of size is the result of the chromonema of these chromosomes, which divide normally but do not separate. Therefore, they remain together to become large in size. The fibers of band and interband regions looks similar in appearance, but the arrangement of fibers in band exhibit a considerable degree of metaphase-like folding and are much more tightly packed. When observed under microscope, Polytene chromosomes show dark and light bands because along each chromatid strand, some regions of chromatin are tightly coiled and other regions are less coiled. At the time of larval development, specific areas on Polytene chromosomes become uncoiled, forming localized regions called puffs. These puffs contain RNA and protein and represent regions of active RNA synthesis. Puffing is due to the uncoiling of chromosome fibers, which are generally closely folded or coiled in the dense band regions. These fibers then project in the form of loops. Their appearance depends on the stage of larval development and controlled by certain specific genes. It is probable that the metabolic activities required for the formation of puffs are related to the secretory function of the salivary glands. A Balbiani ring is a large chromosome puff. These rings are formed by the lateral stretching of loops caused by chromonomata. These loops of chromonomata make up Balbiani rings and give the chromosome a fuzzy outlook. These Balbiani rings are rich in DNA and mRNA, and their formation and function are similar to the puffs. Morgan chooses chromosomes from the Drosophila larval salivary gland for demonstrating the relationship between chromosomes and the heredity information because of their unusually large size and clearly visible banding. The large size of these chromosomes results from a typical cell cycle in which they undergo 10 rounds of DNA replication without intervening mitosis. Major function of the Polytene chromosome is to carry genes which ultimately control physiology of an organism. In Drosophila melanogaster, for example, the chromosomes of the larval salivary glands undergo many rounds of endoreplication to produce large amounts of glue before pupation. Polytene chromosomes may have metabolic advantages as presence of multiple copies of genes leads to high level of gene protein expression. Chromosomes also help in protein synthesis indirectly. Nucleolus contains RNA, and this RNA serves as a means of transmission of genetic information to the cytoplasm, leading to the formation of specific protein. Lampbrush chromosomes are a special form of chromosomes found in the growing oocytes of most animals except mammals. Lampbrush chromosomes were discovered in salamander egg cells by Fleming in 1882. 10 years later, LBCs were identified in shark egg cells and introduced by Ruckert as Lampbrush chromosomes. LBCs occur in many species including vertebrates, invertebrates and even the single-celled alga Acetabularia, but they are best characterized from amphibian and avian oocytes. Lampbrush chromosomes are intermediate structures present during the first meiotic division. These are the largest known chromosomes and can be seen with naked eyes. During first prophase of meiosis, fine lateral loops arise from the chromomeres, and this is the characteristic feature of Lampbrush chromosomes. These loops give it a brush-like appearance. That is the reason behind the name Lampbrush chromosomes. The chromosomes are greatly elongated diploidine bivalents, sometimes reaching lengths of a millimeter or more. LBC's length ranges from 400 to 800 millimeter, which makes them up to 30 times larger than their mitotic counterparts. At the diploidine stage of meiotic prophase 1, chromosomes transfer into Lampbrush form due to active transcription of many genes. The chromosomes transform from a compact telophase form at the end of the last oogoneal mitosis and convert into Lampbrush form, then back reverse into perfectly normal first meiotic metaphase bivalents. These chromosomes are highly extended meiotic half bivalents, each consisting of two sister chromatids. Each chromatid is composed of alternately positioned regions of condensed inactive chromatin and side loops of condensed chromatin. Lampbrush chromosomes are clearly visible even in the light microscope, where they are seen to be organized into a series of chromomeres with large chromatin loops extended laterally. These lateral loops are visible under conventional light microscope due to the coating of ribonucleoprotein. Lampbrushes are extremely delicate structures, and no further progress beyond the pioneer studies of Fleming and Ruckert was possible until a technique could be devised for dissecting them out of their nuclei and examining them in a life-like condition separated from the remainder of the nuclear contents. It consists of longitudinal axis formed by a single DNA molecule along which several hundred bead-like chromomeres are distributed in a linear fashion. From each chromomere, two symmetrical lateral loops emerges which are able to expand or contract in response to various environmental conditions. In the lateral loops, about 5 to 10% of the DNA is present. Lampbrush chromosomes can be dissected in totoform oocyte nucleus. Individual chromosomes are liable to stretching. With extreme stretching, chromomeres begin to separate transversely into two halves so that the paired loops form double-stranded bridges. The axis between chromomeres is also double, which can be seen in certain special regions where two elements separate longitudinally and bear single loops. There is fundamental similarity in the organization of amphibian Lampbrush chromosomes and dipteran's giant Polytene chromosomes. In both cases, very long single fibers correspond to single chromatids and are partly but not completely extended. Substructure of the salivary chromosomes puffs also bear some similarity to that of the Lampbrush lateral loops. Due to their enormous size, Lampbrush chromosomes are something special for the cytologist. They are transitory structures that exist during an extended diploidine of the first meiotic division. Their most conspicuous feature is widespread RNA transcription from hundreds and in some cases thousands of transcription units that are arranged at short intervals along the lengths of all the chromosomes. In these senses, it has been possible to exploit Lampbrush chromosomes in the study of chromosome organization and gene expression during meiotic prophase, and in studies of the molecular and supramolecular morphology of RNA transcription. LBCs provide an ideal system to study transcription, RNA processing and other general features of chromosome organization. Synthesis of RNA. Function of Lampbrush chromosomes involves synthesis of RNA and protein by their loops. RNA is synthesized only at the thin insertion and then carried around the loops to the thick insertion. There it may be either destroyed or released into nucleus. Formation of yolk material. There are some probabilities that Lampbrush chromosomes help in the formation of certain amount of yolk material for the egg. Cytogenetic function. Lampbrush chromosomes of various species have a very similar structure and perform the same function. Comparative studies of LBCs in various species have shown that the side loops seem to be much longer in species with higher C values. Genome size refers to the haploid set of chromosomes. This regularity explicitly reflects differences in the organization of genome sequences. Polytene is a special nuclear differentiation reported in larval and adult Diptera, Collembola, Protista and Angiosperm ovular nuclei. In not all cases, however, do desecrate giant chromosomes represent homologous pairs as in the salivary gland nuclei of Drosophila or Chironomus larva, and in Calyptratae a degree of homolog separation and dissociation of the constituents result in a reticular chromosomal structure. Lampbrush chromosomes are intermediate structures present during the first meiotic division. In a prolonged diplotin state, they undergo decondensation that results in the production of very large chromosomal structure. LBCs are considered as model structure in the studies of transcription control. Changes in transcriptional activity are reflected in changes in their morphology. Transcriptional activity of LBCs is directly connected with physiological process of the body and shows in the morphological structure of the chromosomes. The use of cytogenetic techniques and in situ hybridization have made it possible to identify unique and repeating sequences as well as DNA replication proteins in LBCs.